first of all, let's start with biogenesis of miRNAs. There are two main processes that lead to mature miRNA formation in animals. In the first, you've got nascent miRNA transcripts, or pre-miRNA, pre spelled P-R-I, and those are processed into roughly 70 nucleotide precursors called pre-miRNA. Slightly confusing, this one's spelled P-R-E, not I. And then the second event is that this second precursor, the pre miRNA, is cleaved to generate between 21 and 25 nucleotide long mature miRNAs. Very little is actually known about the transcriptional regulation of pre-pri miRNAs, except that some are located within introns of host genes, including both protein coding genes and non-coding genes. So there might be some transcription regulation through the host gene promoters, but very little is known about this. In addition, you've got certain miRNAs that are clustered in polycistronic transcripts, indicating that these miRNAs are coordinately regulated during development. So, as I mentioned, you've got sequential cleavage of the miRNA precursors, and that's catalyzed by two RNase 3 enzymes, DROSHA and DICER. Both are DS double-stranded RNA-specific endonucleases, and they generate two nucleotide-long overhangs on the 3' end at the cleavage site. So let's look a little bit more at DROSHA, D-R-O-S-H-A. So DROSHA is predominantly found in the nucleus and is made up of the following domains. So two tandem RNase 3 domains, a double-stranded RNA binding domain, and an amino terminal segment, which, well, we don't know what the function is yet. Incidentally, the two tandem RNase 3 domains need to be dimerized to form a functional catalytic unit, catalytic site, in order to be able to cleave the RNA. And then, regardless of the diverse primary sequences and structures of the pre-miRNA, DROSHA then cleaves them into approximately 70 base pair long pre-PRE miRNAs, and this forms the imperfect stem loop structure that I mentioned earlier. The precise mechanism that DROSHA uses to discriminate miRNA's precursors is not known, but several studies have shown that there are features which are important for DROSHA cleavage. So in particular, efficiency of DROSHA processing depends on terminal loop size, the stem structure, and the flanking sequence of the DROSHA cleavage site, because it's been shown that shortening the terminal loop, disrupting the complementarity within the stem, and removal or mutation of the sequences that flank the DROSHA cleavage site significantly decrease, if not reduce entirely, the DROSHA processing. siRNAs are either introduced as a double-stranded trigger directly into the cytoplasm, so in which case it's not processed by DROSHA. So this would be, for instance, if you've got a virus trying to insert its DNA. If it's stably incorporated into the genome, it's possible that it's processed by DROSHA, but I don't really know. I couldn't quite find that one out. Processing is likely to be similar to miRNA, but one of the main differences is that the two strands that form the stem are perfectly complementary, barring obviously random mutations. But this differs from miRNA, which has a imperfect complementarity. So let's come on to how it's exported from the nucleus. after it's initially cleaved, after the miRNA is initially cleaved by Joshua, the pre-miRNA miRNAs are exported from the nucleus into the cytoplasm by exportin 5 or exp5, a RNA-GTP dependent nucleocytoplasmic cargo transporter, basically kind of a little lift, I suppose, that moves from the nucleus to the cytoplasm. Once in the cytoplasm, the hairpin precursors are then cleaved by dicer into small, imperfect, double-stranded RNA duplexes. That's for the miRNA. Obviously, double-stranded, perfect duplex in the case of siRNAs. So this duplex contains both the mature miRNA strand, which acts as a sort of guide strand later on, and it's the antisense part, and then the complementary strands, miRNA asterisk, is also known as the passenger strand, and it's the sense part of the stem. So let's look a little bit more at dicer this time. Dicer is formed of a putative helicase domain, a Duff 283 domain, and a Paz domain, standing for Peewee, Argonaut, and Zvilla. And this Paz domain carries a variant of the OB fold, a module that allows low affinity interactions with the 3' end of a single-stranded RNA. So once you open the trigger, the double-stranded DNA, unwind that, you get single-stranded RNA. And this is what this module allows interacts with. So this acts specifically with the double-stranded RNA that presents the two nucleotide 3' overhang, i.e. those from Joshua cleavage. The other domains that are found in dicer are two tandem RNase 3 domains. So just like Joshua, these need to be dimerized to form a functional catalytic unit to cleave the double-stranded RNA. And it's also got a double-stranded RNA binding domain, or ds-RBD. In addition, you need to have the overhang and a minimal stem length for there to be efficient dicer cleavage. And when that happens, you've got mature miRNAs that range from 21 to 25 nucleotides. This is due to the presence of bulges and mismatches in the pre-miRNA miRNA stem. The maturation of the mRNAs occurs differently, actually, in plants and animals. So in plants, you find that, in plants, miRNAs are produced from long primary transcripts, so just like animals. But unlike animals, we haven't found any Joshua homologue in plants, or at least not yet. We have, however, found 4 dicer homologues in Arabidopsis thaliana, including 2 that may or may not be located in the nucleus. So it is very possible, and some lines of evidence point to this, that maybe at least one of the dicer homologues is specialized in performing animal Joshua's function. So you might be able to explain the diversity of plant siRNAs via the different dicer enzymes. Plants generate 2 classes of siRNAs, so 21 to 22 base pair longs and roughly 25 base pair long siRNAs. In flies, there are 2 dicer homologues, dicer 1 and dicer 2, very, very imaginative names. And deficiency in dicer 1 disrupts the processing of the pre-miRNA miRNAs, whilst loss of dicer 2 affects the production of siRNAs, but not miRNA maturation. This is consistent with the fact that the Paz domain, the pre-miRNA domain, is only present in DICER1 but is not found in DICER2. Both DICER1 and DICER2 seem to function downstream of miRNA and siRNA production to facilitate the risk-mediated gene silencing. DICER2 forms a complex with R2D2, love the Star Wars reference, a double-stranded RNA binding protein, which and together R2D2 and DICER2 are known as the risk loading complex, or RLC. So this complex then associates with the siRNAs and enhances sequence-specific mRNA degradation mediated by the risk complex. So it seems that DICER might have more functions than it was previously suspected, and DICER could very well be involved in both the initial processing of miRNA or siRNA, and then the effector steps of the silencing process.